Enterococcus is a gram-positive cocci. It survived the harsh condition and normal flora of GI and genital tract of the human. 80 to 90% of a clinical isolate encounter the Enterococcus faecalis, and while the remaining majority is made of Enterococcus faecum, and both of these organisms are non-motile. There are some other organisms in this group that is less frequently encountered in the clinical specimen, that is Enterococcus gelinurum, Enterococcus caeciliflavus, Enterococcus avium, and Enterococcus rafinosus. One of the three leading cause of the nosocomial bloodstream infection, urinary tract infection, and rarely isolated from central nervous system and respiratory infection, especially in elderly and immunocompromised host. The organism is also capable of showing resistance to vancomycin. So VRE can be categorized as per the genes, the VAN A, VAN B, and VAN C. VAN A gene is mostly present in Enterococcus faecum, and this is predominant gene. And this gene, the vancomycin MICE strip, show complete resistance, and vancomycin B or VAN B is more resistant because the gene is carried on the plasmid or transposomes jumping gene. The MIC is 32 micron per ml. It is mostly found in the Enterococcus faecalis. When C is found in the Enterococcus gelinerum and caeciliflavus, MIC is 16. The large zone is there on E strip of vancomycin. Selective media for this organism is Campylobacter agar, and selective chromogenic media with the vancomycin is used in the rectal swab and feces. And the organism is sometimes alpha hemolytic or gamma hemolytic. Some strain possess the group D antigen, and widely temperature ranges from 10 to 42 degrees centigrade. Hydrolyzed the bile is choline, NaCl 6.5, PBR and LAP. PBR is pyridinol aralaminidase. LAP is leucine aralaminidase. Enterococcus caeciliflavus produce the yellow pigment non-hemolytic colonies on the blood agar. The pigment can also be used to identify these certain species. If we differentiate between the Enterococcus faecum and faecalis, the faecum is not gamma hemolytic, and faecalis is alpha hemolytic. And the faecum colonies are wide on the blood agar, while as compared to the faecum, the faecalis have smaller colonies with defined opaque edges. Vancomycin dependent Enterococcus strains only grow in the presence of 10 microgram of vancomycin per ml, but upon the subculture on the blood agar fail to grow, the strain isolate mostly from the rectal swab. Arabinose utilization can be used to separate the Enterococcus faecalis and faecum, while the arabinose is positive for the Enterococcus faecalis and negative for faecum. Acidification of a methyl alpha D-glycopyronoside is also used in the identification of Enterococcus species. Enterococcus gelinerum is positive and caeciliflavus is negative for the acidification of methyl alpha D-glycopyronoside. If we have gram-positive cocci, that is catalase negative, bile is choline positive, NaCl positive, arabinose negative, that is Enterococcus faecalis. If arabinose positive, then it may be Enterococcus faecum, gelinerum, caeciliflavus. Then we use the methyl alpha D-glycopyronoside. Methyl alpha D-glycopyronoside is negative for Enterococcus faecum and positive for gelinerum and caeciliflavus. While gelinerum have ability to produce the yellow pigment and it is non-motile and the only species in the Enterococcus family, that is motile, and non-pigmented is Enterococcus gallinarum. We can differentiate Enterococcus family by one another biochemical chart, in which the arginine is used as a basic biochemical test. If the arginine is positive, pyruvate is positive, mannitol is positive, and raffinose is positive, that organism is considered Enterococcus caecilifelis. If arginine positive, pyruvate positive, and mannitol positive, but raffinose is negative, that is Enterococcus faecalis. Same like if the arginine is positive and the mannitol is positive, and if arginine is positive and pyruvate is the positive, then mannitol negative species are further differentiated on the basis of raffinose. If raffinose is positive, that is Enterococcus disparate. If raffinose is positive negative, then it is Enterococcus faecalis variant. If we can examine this chart, we can find out that this entire group can be named as pyruvate positive organism, which is further differentiated on the basis of mannitol and raffinose. Now, among the arginine negative species, which is further tested for albinose and raffinose, which